All right, good afternoon, everyone, and thank you for joining us for the Water Ambassador webinar program. The Water Ambassador program is a partnership program between Martin County and the University of Florida IFAS Extension Florida Sea Grant Program. This is the upcoming set of webinars that we have for our monthly webinar series. So in April, we're going to be talking about oyster aquaculture, a dual approach to building clean water and local economies. In May, we have a panel presentation and Q&A session talking about Martin County land acquisition and watershed restoration efforts. And then in June, we have from Martin County, Carly Batts is going to be talking about stories from the landscape, putting a UF water saving study into practice and the amazing results. And I have to apologize for Carly in the email that was sent out with the links. I had a typo to her title. So I do want to acknowledge that and apologize. It was my error, not hers. I am also excited to announce that we have a rescheduled date for our field trip to the C44 reservoir and stormwater treatment area. Unfortunately, our February date was rained out, so we have a rescheduled date. You can register for the event using the QR code right now on your phone, or I will be putting a registration link as well as the registration link for all of the webinars in the chat. So you can sit tight and hopefully be able to join us on April 9th to visit the reservoir and stormwater treatment area. All of our webinars are recorded and hosted on the Martin County University of Florida IFAS Extension YouTube page. I will also put the link to the YouTube page in the chat. And as a reminder, please put all your questions in the chat box. We will hold all questions to the end of the presentation, at which point we will open it up. I am on my own today. Vincent is in a conference, so just be patient with me. It might take me a little bit longer than it normally does to put all of those links in the chat, but they will all be coming, I promise. So I am excited to introduce our speaker for today's webinar series, Mr. Michael Yustin. Michael is going to be talking about the Martin County Seagrass restoration projects. Michael is a senior project manager at Martin County with 20 plus years of experience managing natural areas and overseeing high-profile environmental restoration projects. A couple of these projects we've actually visited as part of the Water Ambassador program through those field trips, and they're fantastic projects. Great work. Michael's work includes restoring rare coastal upland plant communities, freshwater wetland systems in the Loxahatchee River watershed, mangrove and seagrass habitat in the Indian River Lagoon. He also works to expand public access through the installation of parking facilities, trail systems, paddle launches, boardwalks, and educational displays. Michael will be joining us today, but he won't have a camera on, so take a good look at his face so sort of get the face to the voice as he's presenting with you today. At that, I am going to stop my share and turn off my camera, and I'm going to turn it over to Michael.
Good morning, everyone. Hope everyone is well. I'm glad to be here with you today. Just bear with me a second while I get this all set up, and then we'll be ready to go.
Perfect. Looks great.
All right. Well, as she said, I'm Michael Yustin. I'm Senior Project Manager for the Martin County Environmental Resource Division. I've been working for the county over 20 years, and I've always kind of had a real passion for trying to restore our special places that we have in Martin County. And the newest area that I've been focusing on is in terms of seagrass restoration. So why do we care about seagrass in the Indian River Lagoon? Well, it really is one of the main keys to biological diversity. It's the basis of our estuary food chains and hosts just a tremendous assortment of different fish and benthic organisms, helping make the Indian River Lagoon the most biodiverse estuary in the United States. And historically, when you're talking about the entire Indian River Lagoon, there was roughly 71,000 acres of seagrass habitat. So it's very interesting. And here in Martin County and within this region, from 2005 to 2010, we had this kind of historic drought in this region. And we saw this tremendous growth of seagrass throughout all the areas that you would find it historically. I remember hearing people say that it was as good of seagrass habitat as we had since the 1950s, really. And we had these great, beautiful, lush meadows. And this area that you're looking at is just north of the St. Lucie Inlet in Martin County. And that's typically where we had our best meadows. But we then had this period of decline, such that between 2010, really 2012 and 2018, we lost almost our entire coverage of seagrass in these high class meadow areas. And you can see the difference between the areas on the left, which you can see those seagrass areas, those darkly colored areas in the water, and on the right where there's basically barren sand and very, very little seagrass coverage. Ultimately, this led to this big manatee mortality event that we had in 2021, where something like 1,000 manatees died throughout the Indian River Lagoon. And the reason why they died is because of starvation. They come out into these coastal estuaries during the winter months when it gets cold, and they just had no food. So that really kind of kick-started an interest in restoring seagrass habitat throughout the Indian River Lagoon, because it was a problem that occurred throughout the entire lagoon. So what were the reasons behind this decline? Why did this happen? Well, first of all, between really 2011, 2012, and 2017, we had this big increase in rainfall levels over time, which increased stormwater runoff. And we had a major hurricane, Hurricane Irma, in 2017 that came right up through the middle of the state of Florida, but brought hurricane force winds into our region. And the reason why this was a major factor is because it had effects on water quality. So we started to get more stormwater runoff. You started to get more turbid conditions. You have more nutrients that fed algal blooms, and that resulted in a reduction in water clarity. And if you think about seagrass, it is in fact a grass. It's like a grass that you have on your lawn. It needs sunlight in order to photosynthesize and survive. So if you do things that cause that water clarity to decrease, then you're negatively also infecting the seagrass. So, it's not the nutrients itself, like nitrogen and phosphorus, that are bad because, it's a fertilizer, but it's bad because of it causes competition from algae and other sources. And that is what's driving a lot of this decline in seagrass. So we had this increased stormwater runoff from all of our local basins. Here's a picture of Sandsprit Park. You can just see that water kind of looks like tea or coffee, just barely any visibility. You couldn't see your hand if you put it a foot underwater. And that really was a big causative factor in terms of the declines. But in addition to that, at least in terms of our local estuary, we also had these lake releases, and there were several of them that occurred between 2013 and 2018. And these releases, when they happen, especially on the scale and the timing that they happened, can just really wipe out this area overall. So they were incredibly destructive, and they were definitely, at least in terms of our coastal estuary, a big factor in the decline. However, there was seagrass decline in northern estuaries that occurred as well, which aren't in areas that would have been affected by lake water. So one of the things I've noticed is that there's all these resources that are being poured into seagrass restoration since we had that manatee mortality event. But a lot of these are focused on just plantings and increasing nursery capacity and making it so that we can try and replant these areas. However, we haven't dealt with the underlying problems that cause the loss of seagrass in the first place. So if you do that and you're planting and you haven't fixed the problem, then you're still going to have the problem. So in 2012 22, I first started to become involved in seagrass restoration in this area. And, we decided to focus on this area that was just north of the St. Lucie Inlet, because once again, it's where we had our best seagrass meadows. If you went out there in 2010, you would have seen 100% plant cover. You would have seen 1 to 2 foot high seagrasses throughout a big chunk of this area. And partially that's because, one, there's excellent tidal flushing in the area. So you're getting a lot of oceanic water that comes in, nice, clean, clear water, and that creates good conditions for seagrass to thrive. In addition, it's more of a shallow water area. So it's less than a meter, like less than 3 feet deep. So really, those are the ideal conditions to have lush seagrass meadows. So when I started working on the county project in 2022, and I took really just that first year, and I just kind of looked and try to learn about our local estuary and figure out what we would need to do for a project. And what I wanted to do is I wanted to have a holistic approach. I didn't want to just look at seagrass plantings. I wanted to look at what factors had caused the loss of seagrass in the first place. I wanted to look at both water quality and what's happening in the sediments, because I thought that both of those would be big factors in terms of determining whether or not we have a healthy estuary. And I wanted to then use that information that we gleaned to try and develop strategies that would lead to more successful restoration projects in the future. So what we ended up doing is we decided to look at a bunch of different factors. We wanted to look at everything from rainfall. We wanted to look at water clarity, so how much light is reaching where these seagrasses exist. We wanted to look at nutrient levels. We wanted to look at dissolved oxygen, of which is actually a very big factor in terms of determining the health of these areas. So are we dealing with high or low dissolved oxygen levels is important. We looked at hydrogen sulfide in the sediments, salinity, temperature, and this is actually something I'm going to talk about a little more. We have this benthic cyanobacteria. I call it Lingbia here, but the name has actually been changed to Dapis. And so I'll have some slides that deal specifically with that. And we wanted to correlate it all together. And so the WQ2, 3, and 4 were the prime areas where we're kind of gathering that data. So then in 2022, this funny thing happened, right? Because originally I was actually, and we still are looking at doing seagrass plantings. So we started to go out and we started to look in the estuary, and we were doing this in the late spring to summer of 2022. But this crazy thing happened. We had seagrass everywhere. Any of the areas that we could have possibly thought to plant had seagrass. We were actually, like, me and my counterpart, his name's Patrick Zolwega, is a research scientist from Tetra Tech, and he's kind of my partner on this project. We're going around looking for places to plant seagrass, and we can't find anywhere because all the places that you want seagrass had seagrass. So what was happening at the time is we had this kind of early summer rainy season drought in our estuary, where it really took until late July to August to really start getting the summer rains that we tend to see in May and June. So it created just, you have lots of oceanic water coming in. You didn't have stormwater runoff, and the seagrasses just blew up and took off. Unfortunately, they didn't make it through the entire growing season, and by October, a lot of those increases were gone. And seagrass, it's funny that there is especially shoalgrass, there's kind of an up and down nature to it. It tends to senesce somewhat in the winter, but not to the level that we were seeing in 2022. So the first thing we did is that got us curious. So there's been this debate in terms of how seagrass can grow back into an area. Some think that it's from fragments of seagrass breaking off and moving to a new place and then settling out and then new plants form. That certainly could be a part of the picture, but the speed with which this area came back made us think, well, there's got to be some seeds in the system. So we wanted to know, basically, if conditions were to improve, if the estuary would take care of itself, if the seagrass would start to come back on their own, at least in parts of the system, so that plantings would actually need to be, would not be necessary. So we went out, and this is kind of these research plots that we set up, and we took cores and looked for seeds. And sure enough, we did find some. We found primarily Halophilia decipiens and Halophilia johnsonii. Our work was pretty limited, though. We didn't, and we also correlated that with mapping of seagrass to see how they correlated together. And although we found seeds, it was really just a preliminary study. And we're hoping to get funding to expand that study and look much deeper than we did before. I know some of our partners in the northern Indian River Lagoon are actually starting to look at that as well and trying to understand if there are seeds in their systems. And then I showed this on the past map, but we also looked at seagrass. And we went out and we created these big maps to determine what areas had seagrass, what species we were finding, and then what percent cover of those species were in this area. And we tried to look at different areas, different depths, so that we could get more of a holistic, full view of how our seagrass was doing. Oops, I guess I repeated that slide. Sorry about that. So, and I mentioned this, we started to monitor and map our seagrass. And we found this, and we started to do this in 2023. So 2022, we're just planning. 2023, we started to go out and do some monitoring of these areas to better understand what was going on. And we saw this interesting trend that was similar to what we saw in 2022. In March, we saw seagrass was only about 10% covered throughout these areas, but you can see there's this big upward trend such that within really just a few months, it went from 10% cover to, in one area, as much as 60% cover. So we had these big, lush, healthy meadows. We're coming back. We're very excited. We thought things were going well, but then something happened. And it happened between June and August of that year, such that by August, that seagrass coverage was starting to decline. And by November, if you look, it was pretty similar to what it was in March. So then we start to ask, well, what factor could have caused that reversal? And this is where I talk about that cyanobacteria, now called Dapis, was called Lingbia before. And so this is it's a type of cyanobacteria, but it can be both benthic or pelagic. So what it does is it moves into these areas, it will adhere to the seagrass, and then it'll expand through the summer months. And what'll happen when it's really successful, it will actually fill up with air. So air will fill up within the tissues of this, and it makes it buoyant. What ends up happening is it will break or even uproot the seagrass. So if you look at this photo on the top left here, you can see that's some of this Dapis that's floating. You can see little seagrass blades are all mixed up within it. And so it's causing it to uproot. It will carpet the grasses, so it decreases light condition. And it has this funny relationship with dissolved oxygen. And under certain conditions, it can cause anoxic soil conditions. So what we wanted to do, and what we started doing in 2023, is we wanted to do a little experiment. We wanted to see, we took some transects and we started to remove it using this experimental technology. I mean, it wasn't anything complicated, but basically you had a pump and a vacuum, and then our biologists would go through and just try and remove it from these seagrass areas. And then we would monitor both the concentration of the Dapis, and we monitor the seagrass to see if there was a difference in terms of seagrass survival when you were controlling and removing it. And this just is a video showing more close up what this stuff looks like. So I'll just let this play. This stuff, it's pretty disgusting. It's like this green ooze or goo. We called it the schmutz, was the word that we came up for. But it's, you can just see from this M frame picture here how much it's overtopping the seagrass. You can see there's little green blades sticking through, but you can really tell how it's competing with that seagrass and how it would negatively affect it. So then what we did is we went and we compared the Dapis lyngbya with seagrass. And we set up these monitoring transects. And you can see this relationship right here. So your bottom line is your seagrass percent coverage, right? And you see it's going up and up and up. But then between June and August, you can see it dropped. And at the same time you see that drop, you see this massive increase in lyngbya percent coverage, so that it was about 90% lyngbya coverage. It went from about 30% to 90% in 2 months period of time, right at the same time where you have this massive die off of seagrass. Then you can see by September, there's still a lot of it. It's about 50 to 60% coverage, but the seagrass is continuing to decline. And by November, it's completely gone. And you see that trend repeating with it. So you can see it in this other area where it just blew up, went from, once again, about 30 to 90% coverage. And then you see just this big drop in terms of seagrass coverage. So that brings us to kind of the next phase of our analysis. So we did a lot of water quality sampling, and we wanted to look at dissolved oxygen. We set up these hobometers, these sensors, and they were placed at the bottom, right above the sediment surface in the areas where we had the seagrass, and we were measuring things like dissolved oxygen, light, salinity, and temperature. And then we took that data and we correlated or compared it against our seagrass monitoring data. And this is one of the interesting results that we found. So this is from 2023. And what you're going to notice here is the blue line represents the amount of light. I won't go into kind of the details, but it's the amount of light that was apparent at the sediment surface. So the higher this is, the better, in essence, for our seagrass. But you'll start to see that you start having this decline occurring in the amount of light in our areas. And it's interestingly, so this red line represents rainfall data. So this is a 0.5 inch, this is 1 inch. So in April, we had this 3+ inch rainfall event. And then look at like the light data starts to go off a cliff. So obviously rainfall, stormwater runoff is having some effect in terms of the amount of light that's present in these areas where our seagrasses are being found. And here's another interesting trend that we found. So the orange line that you see here is temperature. This blue line is your concentration of dissolved oxygen in the water column. So you're going along, this is March into April, and see you have this kind of nice tight range in terms of how much dissolved oxygen you have. That's actually good. It's healthy. It never drops down to zero, and it never gets too high. But look what happens in April. You start to see these big, massive swings, and they correlate really well with when your rainfall event's happening. And this continued throughout the growing season. You know, you're not meant to have like these big, huge peaks of dissolved oxygens, and it's bad when it's dipping down to zero in these areas as well. So this relationship is specifically indicative of the cyanobacteria. Seagrass will play a role as well as a respiring plant. You know, it produces dissolved oxygen. So seagrass will cause increases of dissolved oxygen during the day, and it will drop at night when the seagrass is not photosynthesizing. But when you add Dapis to the mix, it just takes that data and puts it on steroids. So you get these mass fluctuations, really high peaks during the day, really low valleys at night. And in addition, you think about it, if you get several days where you have cloudy weather, right, you're not going to get those peaks anymore because the Dapis is not going to photosynthesize. But it will tend to suck the available dissolved oxygen out of the water column at night. We think that specifically played a role in terms of this big decline that we had. So I'm not going to go too far into this. This is a little technical, and we actually need to do a lot more work on this. But one thing we did look at is we wanted to look at lake releases and that they affected our water quality variables, but also looking at how things trend together. So this is a multivariate statistical analysis. And the only thing I'll say about it is when you have groupings, they show relationships between different things. So you can see on this, on the top end, you have average cyanobacteria, average dissolved oxygen, and average seagrass. So they tend to all come together. In addition to that, there's an association between salinity, nitrogen, and phosphorus. We did, not only did we do water quality testing, but we actually did pore water testing where we would draw samples from out of the sediments to look at different variables, nitrogen, phosphorus, and dissolved oxygen. But there was a close relationship between nitrogen, phosphorus, sulfide levels, and salinities. So to me, that means once you're getting more stormwater runoff, you'll have lower salinity levels and you'll have higher nitrogen phosphorus ratios, so on and so forth. And this is pore water phosphorus and nitrogen. So those two tend to be closely related together. And we did this over a 12-month period. So what is the reason for this relationship? Why are we seeing this kind of relationship in the estuary? Well, first you have these rainfall events that start, once we get our summer rains coming through, and once those rains accumulate enough, you start to get stormwater runoff. So all of our stormwater areas, or all of our town, city areas, that water from suburban and urban agricultural areas starts to get washed into our water bodies, that gets carried downstream, and eventually those nutrients make their way into the Indian River Lagoon, and they feed these Dapis blooms. And one thing I will say, literature has shown that there's a relationship between nitrogen specifically and Dapis. So nitrogen, actually the main source of nitrogen in our system, tends to be septic systems. Agricultural areas tend to produce more phosphorus. And this Dapis has been shown to be a nitrogen fixing organism. So there is most likely, and we haven't proven it definitively, but there's a good chance that septic systems and human waste are the primary driving forces behind these blooms of Dapis that we tend to see. And in addition, once again, it produces oxygen during the day and consumes it at night. Which brings us to our next part. So one of the reasons why low dissolved oxygen can be a problem, especially when you combine it with poor light conditions, is that the thought is that poor light conditions stress the grasses out. And when you remove dissolved oxygen from the area, you tend to get a lot of hydrogen sulfide production. So basically, the hydrogen sulfide in the sediments then becomes more of a problem for the seagrass than it would be if you had healthy seagrass and you had enough light to the area. So you're taking seagrass that's already stressed, and then you're adding in this hydrogen sulfide in the sediments. And that is what we think proves toxic to the seagrass. So I saw this presentation from this professor. She used to be at University of Florida. Her name is Diana Chin. And she had done this research on this particular family of clams called leucinid clams that are often found in seagrass meadows. And what she found in her experiment is that these clams tended to improve survival under a mesocosm, under an experimental setting, when she put these clams into the mix and she stressed these seagrasses out, that the seagrass in areas that had these clams have better survival and better health indicators than ones where the clams were not present. And she also measured hydrogen sulfide in the sediments in the system. And what she found is that the hydrogen sulfide levels were lower when these clams were present than if not. And the reason why that happens is because these clams, this family of clams, they have a bacteria that's associated with their gills, and it's a symbiote. This bacteria actually needs and uses hydrogen sulfide. They consume it, and in doing so, it actually powers the reactions that are needed for the clams itself. So basically, if you add these clams to the mix, this bacteria will help to reduce this hydrogen sulfide levels. That leads to a healthier estuary. However, the thing about these is this family of clams, they've never been grown in an aquaculture setting before. And we saw, Diana saw that they have value and that they would be good for helping to restore seagrass, but you can't just collect them from one area and bring them to another. You have to be able to mass produce them. So what we wanted to do is we wanted to then take those clams and try and care for them in an aquaculture setting. And we actually went out into our local estuary and we found one particular species. They're called Divalinga quadrisulcata. You can see these guys are tiny, like they're like fingernail size. But they live, and there's a good number of them in our coastal estuary. And so what we're working on now is with a group called Great Florida Shellfish Company. They're housed over at Florida Oceanographic Society, and they have several sites across the state. So what they're trying to do is they're trying to, one, be able to control them or be able to support them in this aquaculture setting. So you collect them, you care for them, you see if you can get them to grow. And the ultimate goal is to try and spawn them so that you can produce millions of them that we could then just distribute it out through our local estuary. So we went and collected these clams. They brought them back to their facility. What you're seeing there, it's called a strip spawn. So they'll actually crack open the shell and look for sperm or egg material. And it's kind of a simple thing. You then just pour them into a beaker. You take some of the sperm and you mix it with the egg and you hope for fertilization. And then, hopefully millions of baby clams. So we've spent a lot of time first learning how to care for them. Now we're working on the spawning side. We've attempted to spawn 3 times. We have 1 success and the other 2 times we're not. And we think the times when we were not is just because of the eggs weren't ready. So there's a particular time when the eggs will be ready. And once that happens, they're receptive to the sperm and you can have reproduction. So we're still trying to better understand how to spawn them. We're getting closer. But once we do, then you work on the next phase, which is growing them from when they spawn in order and until such time as they're big enough that they can survive in our local estuary, and then you'll release them. We're hopeful within the next 6 months to a year to really have a solid grasp on this. And then the cool thing is that this will be available to resource managers throughout the estuary as another tool in their toolkit to try to improve the health of our local estuaries. In addition to that, you could then look at other species from the lucinae family. And my recommendation is you kind of look at your local estuary, you find what lucinids are in that local estuary, and those are the ones that you then take and try and work with in an aquaculture setting and release into that area. So I know I kind of badmouth seagrass plantings a little bit, and I didn't necessarily mean to do that. I do think that they have a place in this whole attempt to restore seagrass, but it should be part of a bigger picture. And since we don't really have solid numbers in terms of our success of seagrass plantings, it should be done kind of experimentally where you try different things. So what we're trying to do is we're trying to plant at different seasons throughout different areas of the estuary, and then just kind of watch what happens and see how successful they are or not. We're partnering with Florida Oceanographic Society. They have a nursery and an area of their game fish lagoon that's actually dedicated to seagrass. So they have seagrasses that are just growing, living in those areas. We collect them and we're taking them out and then doing different plantings throughout our local estuary. And this shows an area, this was taken, I think, about a month after plantings. So you can see little seagrass blades popping up all over the place. You know, it was kind of funny though. So we did this project and apparently we timed it perfectly when there was this big sea hare reproduction event. So we plant all these seagrasses and immediately all the grasses we planted were literally covered with these sea hares. And we're all worried. We're like, oh no, they're just going to eat them all and then they're going to be gone. And sure enough, they did die back to almost the sediment surface, but then the grasses just came back. It was almost like you're just mowing them, and then they spread out throughout the area and the sea hares moved on. So it wasn't like a regular normal thing. Oops. Which brings us to where we're at now. So we're hoping to expand our efforts and do kind of a multi-pronged effort that will include continuing to sample our water quality areas and potentially expand that to look at some new areas. We really want to expand that seed bank study and try to crack the code, you know, in terms of how our seed bank looks in this local area, the estuary. We want to do more benthic habitat mapping and expand that effort, so to really just continue to get a better picture of how healthy our seagrass populations are. In addition to that, we want to release clams out into our area, and not just the leucinid clams I mentioned. We also want to release some Mercenaria campotensis clams. They're a hard clam. They're more associated. They can help improve water quality. So we want to experiment with adding those into the mix. And then on top of that, do some seagrass plantings. And finally, we want to continue, we want to try and upscale our DAPIS removal project. And we're trying to come up with a technology that could do a bigger area than just, you know, Patrick with his little vacuum dredge going along and sucking the stuff up. And that's kind of it. That's our seagrass restoration project. So I guess I'll stop sharing so I can take questions.
Thank you so much, Michael. It's fascinating because we hear so much about restoration plantings and how, you know, they aren't as successful as we hope. So it's really great to see that you're doing the work to try and understand how to make these efforts successful. And it's fascinating work. We have not received any questions in the chat yet, so we'll wait a few minutes. I'm going to allow everyone some time to put some questions in. And in the meantime, I'm going to post our evaluation. But I want to thank you so much for your willingness to present today while we wait to see if anything comes in.
You're welcome. And, you know, with this effort that we have ongoing, it might all fail still. Like, you know, it might be the answer is $10 billion or whatever in water quality improvements to the system to get the pollutants out of our estuary. It's not, this is not an easy fix. It's not an easy problem, but at least, you know, trying to contribute to our understanding of why and how this decline is happening, and provide tools that, you know, even if we get a small increase in terms of seagrass health in our local estuary, that's still a difference and could fuel future improvements.
Yeah, absolutely. So we did have a question come in the chat. Jaden asks, do you simultaneously track how seagrass restoration efforts bring back other benthic or aquatic species that may have been affected by the significant reduction in seagrasses in the estuary? So do you do like any informal work or monitor any other?
No, we don't.
Okay.
And there's certainly lots of unexplored areas in terms of, you know, seagrass health. I know Austin Fox from FIU is really, you know, he's clued in on this whole issue in terms of a decline in your benthic organisms and how that relates to seagrass health. It could go hand in hand, and it's an area that I'm curious at as well. You know, we dig a lot of holes out there looking for these clams, and in a lot of these holes, you're not finding much other things that are living as well. And you would expect that you would find tons and tons of worms and you know, different clams. You know, we find, and I'm not saying that they're not there, and I have nothing to base on that evaluation to say, you know, there should be more of this, but it just is a feeling on my part that we should be finding more than we actually do.
And we've got lots of questions coming in. So
All right.
the next question from Sarah is there any concern that introducing clams could alter the ecology further?
Well, I mean, I think you have to ask that in terms of when you restore anything. I don't see, I actually see the opposite. Like, I definitely feel that, you know, we're in a situation where there's less than there should be. So increasing, and like, I don't see the downside to having an organism in the sediments that can reduce hydrogen sulfide levels. So I understand what you're saying, but I don't, you know, feel that that's a problem in this area at this point in time.
Okay. And Kent asks, it's challenging to produce reducing environment in the sediments to enhance lucid survival. What approach are you using to do this in the mesocosms?
Well, we're not working in mesocosms yet in terms of experimenting. We're really, that was Diana Chin's work, and that's what she looked at. I'm not saying that there shouldn't be, but our goal is once we're able to produce these consistently and we can start using them for seagrass restoration, then another area of focus could be to work with some of the research partners and to start experimenting with them in different ways. Um, you know, and better understanding the association between them and seagrass. You know, another question. So we find lots of dead shells of other types of leucinid clams, but uh, we don't find this. The divalingas are the only ones that we're finding in quantity out in these areas. So then the question then becomes, are these more adapted to surviving without seagrass? I think there's an interrelationship between the two. I think the lucinids need the seagrass and the seagrass needs the lucinids. So that's why co-restoration with different types would be pretty valuable. Did I answer that question or did I get lost?
You got a thumbs up from Kent, so I'm going to
Okay. Yeah.
assume yes.
All right.
The next question from Ariel says, really cool and interesting work. I have lots of questions, but one of them is what impacts, if any, were there to seagrasses using the DAPIS vacuum? Any other removal tools in the works that you can talk about? We have
Yeah.
seasonal DAPIS issues in Charlotte Harbor Estuary as well, and we're exploring options for removal.
Yeah, so the one thing we did notice, that first year, we were only doing we only did the removal every other month. And we did find statistically a statistically significant relationship between removal of DAPIS and seagrass health. But the problem was between June and August, there wasn't a removal event. And by August, the percent cover of the DAPIS wasn't different between the control area and we actually experimented with the removal. So we think that we need to do more frequently in that period between June and October, potentially. So it could be that it's necessary to do once a month. And, you
And
know,
we had, oh, I'm sorry. I was going to say, Ariel has a follow-up of what was the size of the restoration area?
oh, these are just like small transects. So it was like 1 meter wide by 10 meters long. With the technology we're using, it wasn't feasible to do a big area. We have been working on ideas to do that across a wider area. The problem, you know, you want to get the DAPIS. We were actually thinking about coming up with it's kind of hard to explain. It'd be a soft rake that would stay up elevated off the bottom and move slowly, and that would kind of stir it up, and then have an outward facing vacuum that would then suck that material up, you know, because you don't want to be sucking across a big area across the bottom. So we'll have to stay tuned. And I'm actually, we've been trying to get funding, and we haven't gotten funding yet, which we would need to in order to take this another step.
Next question, he says, "Hey Michael, it's Nick from the FOS team. You mentioned that we need to solve the issues in the area before planting. What is being done to solve the septic issue by the county? I was born and raised in Martin County, and it's always been an issue. I know that on the lake side of things, it's a separate issue, and that's ongoing with the Army Corps. But so what's the county doing about the septics?"
The county has been very active in multiple different ways. Not one, also building stormwater treatment areas. So these areas that pick up these nutrients, them flowing into our river system. If you have stormwater treatment areas that capture some of that water and then treat some of that water, then you can remove it from the system. The county has been very aggressive in terms of our septic to sewer conversion program. It's a difficult thing because of it's not cheap to do that. It takes time and it takes a lot of money in order to convert all the people that are needed to be put on sewer system as to help with the issue. And in Martin County, we're also dealing with, and it's not talked about a lot, but a lot of our watershed drainage is actually from St. Lucie County, because you have the North Fork of the St. Lucie River, and there's an order of magnitude larger number of people in St. Lucie County than there is in Martin County. So part of the issue is waiting on their septic to sewer program to get to the point where it would make a difference in our local estuary.
That's a great point, because watersheds don't follow municipal boundaries.
No, they
You don't
don't.
often think about that.
Yeah.
The next question is from Mark Perry. Did you consider if Lyngbia blooms causing the decline in seagrass cover during a non-growing seagrass season?
The Dapis doesn't tend to really grow that much. You saw the one slide, but I know like it tends to be gone between about November and April, at least in our seagrass estuary area. Part of that, and it's one would be temperature would be a factor, but the other is that without the seagrass there, it has nothing to hold on to, and it'll just get flushed around. So as the seagrass dies back, it has less area to grow. It's really, literally the seagrass is a convenient place for it to attach and not get swept out and in with tides and currents.
It's obviously an interesting topic because the questions keep rolling in.
Yeah.
Greg wants to know, what is the natural range of the lucinated clam that you're working with, and are there any studies of it elsewhere in the IRL?
I don't think there's a solid understanding of where lucinids are or are not present. I don't think I know probably Smithsonian, I'm sure, has done some work. If anyone would have that information, it would be us. I would suspect that it's found pretty much throughout the entire Indian River Lagoon. Actually, Diana Chim might have some knowledge about that, but it's certainly, when we went out the first time with Diana, we had no idea where we were looking or where we were going to find it. We literally just picked like 10 places and then just started digging holes, and across a broad spectrum of different areas. And that one area, the flats area off Bathtub Beach near Sailfish Point, for whatever reason, tends to have the highest concentration of them.
Nick Muzia asks, have you and Patrick looked into the N to P ratio and how low nitrogen to phosphorus ratio supports cyanobacteria? Example, sediment samples in the areas of interest.
So we have, we want to form a partnership with Harbor Branch, and the idea is to collect nitrogen and phosphorus data from the Dapis and from seagrass to establish that. We're actually, somebody from Harbor Branch today is going to pick up some tissue samples we collected last fall of the Dapis. So they'll be able to do some N to P ratios on that. So it is something that we want to explore. And in addition, do nutrient isotope tracking to basically that will tell us if sewer system or septic systems is a driving factor behind this or not. So yes, we are looking at that, but the more you look at this, the more questions that come up and areas that you could look.
Yep, and I think that sort of speaks to the next question, which talks about a lot of the nitrogen loading to the St. Lucie estuary. I'm going to paraphrase it for your benefit, but it's essentially the 46,000 plus septic tanks in the St. Lucie estuary produce about 13% of the total nitrogen load. Can you make sure that you include all of the sources of nitrogen loading into the St. Lucie estuary in your analysis and conclusions? Thank you. This is from
I think it would be, yeah, I think it would be hard to go there for this particular analysis. There's only so much one study can look at. And, looking at sources throughout the estuary is just not something in this particular project that we're looking at. There are other groups that are looking at nutrient loadings from the system, and there's people that could speak more to that issue. So I'm not looking at the sources more than just the isotope tracking and understanding where this could be coming from.
Okay. And I think this next question comes in response to your comment about looking for funding to continue the work. So Ian asks, how has the work completed to date been funded, federal, state, local, and/or private? Does work simply stop if proper restoration requires more funding than has been provided?
So the county does have an ongoing investment in seagrass restoration. And really, actually, we're calling it Indian River Lagoon restoration. So the county is putting in $100,000 a year for that. And that can include any of that stuff, from clam restoration, the seagrass plantings, the water quality. But this particular work was funded with $260,000 grant from the Environmental Protection Agency. The county actually got it in 2021, 2022. Technically, it was just to do seagrass plantings, but I kind of inherited that project, and I'm like, oh, I don't want to just do plantings. I want to do it as part of something bigger. So that's funding kind of all the stuff that we've been talking about. We did just get $60,000 from the Southwater Water Management District from the Indian River Lagoon license plate program, which is specific to clam restoration, and that will include leucinid clams and these mercenaria clams. I should mention it's not technically a grant, but when we formed this partnership with Great Florida Shellfish Company, we offered to construct a facility for them to specifically deal with these leucinid clams. So we gave them $30,000, and in exchange, they're matching it with spending their own staff time in doing the work necessary to attempt to understand and grow these animals. And I will say that the amount of, like, if you look at the staff time and put a cost on that the amount that Florida Shellfish has spent far outsees the $30,000 investment that the county made.
Great. And the last comments in the chat, I think, sort of supplement some of the stuff that you've discussed. So Amy Easton, also with Martin County, says that Martin County is in the middle of an integrated water resource plan that will systematically examine solutions to flooding, sea level rise, and water quality as a whole systems approach. And she adds that loadings will be examined watershed by watershed in the plan, starting with land use and septic locations. And then Nicholas adds, FYI, nitrogen is much easier to remove, example, biofiltration and the nitrogen cycle. Phosphorus is much more difficult to remove as it does not go away like nitrogen and through the nitrogen cycle. So I will, with the last couple minutes we have, I'll let you comment to those comments if you have anything to add.
No, I mean, Amy is much more of an expert in terms of that than I am, so I'll defer to her expertise when it comes to that particular program. And there's some, I think nitrogen removal is a little more difficult than it's given credit for. I think one of the things that needs to be looked at is how effective STAs are at treating nitrogen from the system. That's something that the county has been looking at.
Well, thank you so much again for a fantastic talk and some great conversation discussion at the end. Want to, thank you all for joining us for the Water Ambassador webinar series. We hope to see you in April for our next webinar on oyster aquaculture, and hopefully for our field trip on April 9th for the C44 canal. So thank you all again. And Michael, thanks so much for a great presentation. Have a great
Thank you.
afternoon, everyone.